So welcome everyone to our weekly AITRN seminar. The speaker today is Robin Belton from Smith College, and the title of her talk is Time Series and Biological Network Analysis via Directed Graphs.
Yes. Hello, thank you so much for the invitation to this really nice online seminar, and thank you all for being here. So the work that I'm talking about is in collaboration with Bree, Brittany, and Tomas, who are all faculty members at Montana State. And I'll be focusing on the work in the paper Extremal Event Graphs, a stable tool for analyzing noisy time series. And then we do have code for the computations of the constructions that I'll be talking about. So for our biological motivation, we want to compare model organisms to non-model organisms to answer biological questions such as, can the circadian rhythm genes of the mouse provide evidence for an intrinsic clock in a malaria parasite? And then can we use these genes that run the cell cycle in yeast that is used for baking to find genes that run the cell cycle in yeast that causes deadly yeast infections? So we're interested in biological questions about circadian rhythms and cell cycles. And we're going to study these questions through time series data, which is a function that's varying over time. And specifically, we look at multi-dimensional genomic time series data. So we're looking at how gene expression varies over time. And gene expression is the process by which information from a gene is used in the synthesis of a gene product that can be used to produce proteins. And it is measured by either the amount of mRNA or by the amount of corresponding protein. And this can be used to measure the level of activity of a given gene product here. And the data that we're working with is discrete. So it's gene expression taken from a cell over thousands of genes, and it's only taking at a finite number of time points. And so here's just a very simple toy example of two genes, the orange gene and the purple gene. So going back to our question, we want to compare genomic time series data of model organisms to non-model organisms. And we're going to do this by trying to capture the order of the mins and maxes in the collections of time series. And we want to do so in a way that is robust to time measurement noise, since our data is going to be noisy. And the reason why we focus on the order of mins and maxes is because there is strong evidence that the relative phases of oscillating regulators are important in controlling cellular processes such as the cell cycle or the circadian rhythm. And it has previously been shown that the ordering of extrema is a reasonable approximation of control by phase relationship. Okay. So as I mentioned, we're working with noisy data and we want to capture our orderings, but make sure we take into noise into account. So what do I mean by that? So looking back at this example, if we were to look at the data, we would say that this max at S comes before, of our purple max here, comes before the orange max at T. So we would have this ordering. However, if we assume an error level of epsilon, meaning that we can place these epsilon bands around our functions and saying that the true underlying gene expression could lie anywhere in these bands, then we could get the opposite ordering. So for example, for our orange gene, we could get, sorry. Okay, I was remembering purple comes before orange. Okay, so I want orange to come before purple. All right, so for our orange time series, we could get something that looks like this, whereas for our purple one, we could get something like that. And so now our purple max comes after our orange max here, which is the opposite of what happened before. All right. So we want to be able to capture that somehow, and we're going to do so in a weighted directed graph descriptor of our collections of time series data that is robust to this discrete measurement noise and keeps track of the order of the extremal events. And then once that is done, then we want to define a distance between these weighted directed graphs that we call these extremal event DAGs. And we want to do so in a way that can be computed, where DAG stands by directed acyclic graph. And
Robin,
then we're going to use these directed
Robin,
graphs and distances
Robin?
to study the data. Yes.
Question. So do you mean local max by max?
Yes.
Thank you.
Yeah, so we're looking at local maxes and mins. Good question. Cool. Any other questions? Cool. All right, so first is the directed graph construction. And it's going to be computed from a collection of nicely tamed functions from a closed interval into the reals. And what I mean by this is that we're going to have continuous functions with a finite number of local mins and maxes. And we're also not going to have any like plateau type mins or maxes here. So this will be our input. And I mentioned that our data is discrete, but I'm going to describe the construction with continuous functions because it's a little nicer. And we do have a way of going from continuous to discrete where the results still hold. All right. And then our output is going to be this weighted directed graph where we have weights on both nodes and edges. So first, for constructing the vertices is that each vertex is going to represent a local extremum of the function. So here I have one of my favorite functions, sine of x, from 0 to 2 pi. And it has 4 local extrema, so we're going to have 4 vertices. And then for our vertex weights, we want this to be the largest noise level for which we can say the extremum definitely exists. So what is the largest epsilon band we can place around our function and say that under any perturbation of this function, there's definitely a local min there or definitely a local max there. And the way we can compute these values is through sublevel set persistence. Right. So we can just do our standard sublevel set persistence, where we can imagine our function is behind some curtain. And as we raise the curtain, we're going to reveal different connected components, and also connected components are going to merge together. So at first, we don't see anything. At stage 2, we can see our first connected component. At stage 3, there are 2 connected components that are now here. At stage 4, they have merged together into one. At stage 5, another connected component is coming into play. And then at stage 6, they have merged together and we see our entire function. And the key observation is that new connected components appear at heights of local minima, while connected components merge together or die at heights of local maxima. Right. So now we want to record this information, and we can record it in a persistence diagram where the x-axis is birth. So these are the heights at which the connected components appeared, and the y-axis is death. So these are the heights at which the connected components merge together. Right. So each of the birth coordinates is corresponding to a local min. And so we can compute our weights by looking at our diagram and getting weights for the mins. And so here, how we define it is that if we look at our min at T3, that's corresponding to this point highlighted in pink in the diagram. And so we have our birth coordinate of f of T3, our death coordinate is f of T2, and then our vertex weight is going to be the persistence of this point divided by 2, is how we define it. And then similarly, we can travel around our diagram and get vertex weights for the other mins. So for the green one, we do a similar procedure. And then for the min at T1, the death coordinate is infinity, and it's hard to do arithmetic with infinity. So we just do max of our function minus f of T1 divided by 2. So at this point, we have vertex weights for all our local minima. So lastly, we just need to get our vertex weights for our local maxima. And what we do is we just take the negative of our function so that all of our maxima become minima. And then the heights between max and mins, or the distances between the heights of max and mins, remains the same. And so then now we can apply the same process here. All right, so looking at our persistence of our points, divided by 2 will give us our weights for our maxes. All right. So now I have that filled in, and then the next step is adding in the directed edges and the directed edge weights to our graph descriptor. And so for directed edges, we're going to add a directed edge from A to B if the time of event A precedes the time of event B. So we do this so that we're capturing the ordering of when this all happens. And then for our edge weights, we want to find the largest noise level for which the order of the 2 extrema can be discerned. So what is the largest epsilon bands we can place around our functions and say that this min definitely came before this max. And that's what we want our weights to measure. So we have a nice result that if the nodes are corresponding to extrema from the same function, then that edge weight is just going to be the minimum of the node weights. So in this example, I'm starting to construct the extremal event DAG for sine of x shown on the left, but also adding in a cosine of x on the right. All right. So we have those directed edge weights. And then the last edge weights we need are our cross edges between extrema one from sine of x, one from cosine of x. And computing those weights is a little more complicated. And to do that, we use what are called these epsilon extremal intervals here. And I have the definition on the left, but it's best described by picture. So if we look at this, we fix this epsilon, and we look at this max at pi over 2, how we compute it is that we just go down by epsilon. We shoot out until we hit our f plus epsilon, and then we project this down to get our epsilon extremal interval, which we denote as phi epsilon of pi over 2. Okay. And then for mins, we do this symmetric analogous process. So we start here, we go up by epsilon, we shoot out until we hit f minus epsilon, and then we project this down to get our phi epsilon of 3 pi over 2. And the reason why we use these intervals is because they have this nice property where that if we were to take any epsilon perturbation of our function here, say this, then we will be guaranteed to have a max within this phi epsilon of pi over 2, and we're guaranteed to have a min within this phi epsilon of 3 pi over 2. And so using this property, we're able to compute these cross edge weights. Right? And the result is that if we have nodes that represent extrema from two different functions, then the edge weight is the minimum of the two node weights and the smallest epsilon for which we have a non-empty intersection. So in this example here, this epsilon is that epsilon star value, where the extremal intervals around T3 and T4 just begin to intersect one another. And then lastly, returning to our example with sine and cosine, if we're trying to figure out our edge weight between the max of sine at pi over 2 and the min of cosine at pi, then in this case, we can use properties of sine and cosine at their translations of each other and a little bit of algebra to find that this epsilon star value is going to be 0.14, and that will become our directed edge weight. All right, so now that finishes up how to construct these extremal event DAGs from a collection of time series. And now we are ready to talk about distances. So how can we compute distances between these objects? And the idea is that we're going to construct this supergraph so that each DAG is contained in this supergraph. And then the supergraph is also going to contain two weights on each vertex and each edge. And so then applying some LP distance, what we do is we just take the absolute difference of the weights on the vertices and edges and add them all up, and that will give us a distance value. And so this is trying to measure how similar our time series data are and keeping track of order of mins and maxes. All right. Now to construct this supergraph, we use these, what we call backbones. And so for a backbone is going to be the ordered sequence of min and maxes along with their weights. So for this cosine-ish function, we have a max of a weight 1/2, followed by a min with a weight 1/2. And then we have a small max and min that have weights of 0.02, and then our final max of 0.5. And then if we want to compare two different functions or two backbones, then we consider these alignments. And so we want an alignment such that we're matching maxes to maxes, mins to mins, and we're also preserving the order of the mins and maxes. So if we just look at these two functions, f and f prime, we would want to match this max to this max, this min to that min, have this little max and min get matched to these empty nodes or insertions that we call them, and they're going to have a weight of 0 to it. And then we can align this max to this max. So our alignment would look something like this. And then we can also have this backbone distance, which is also a type of edit distance, where we're going to sum up the differences in the weights as our costs. And so for our backbone distance, we want to find the best alignment that preserves order and keeps our labels the same. And then we can add up the differences in the weights. So the backbone distance for here would be 0.02 plus 0.02 or 0.04. Okay. And then just a little bit of what this backbone distance is capturing versus an LP distance is that for these plots, what I did is I looked at the time series for SY4, this gene here, and then I compared it to a bunch of different CNAG genes, like hundreds of them. And what's shown on the left here is that the distance between SY4 and this CNAG gene was the smallest out of the distances between SY4 and all the other CNAG genes. In terms of the L2 distance. And for an L2 distance, we're looking at point-wise differences, and we're trying to minimize these point-wise distances. Whereas backbone, it's really matching more about shape. So we see that, for these two time series here, this Y4 and this CNAG 06818, this produced the smallest backbone distance out of all the CNAG genes. And we can see that they have the same type of shape and similar orderings of mins and maxes and similar prominence of the mins and maxes. However, there's a little bit of a shift, which is why they're not as close in an L2 type distance. So depending on what your application is, it may be better to be using an LP distance, or maybe it's better using a backbone distance. For example, if you know that all your experiments aren't always aligned or starting at the same time, then maybe you want to use a backbone instead of LP. Okay. And so now we use these backbone distances to build up to this extremal event DAG distance in this super graph here. And what we do is we first assume that there is a one-to-one correspondence between the functions in our two data sets here. And so for here, we could align this cosine function to this cosine-ish function over here. And then similarly, we would align this sine-ish, have a correspondence between the sine-ish functions. So then we would first compute the backbone alignment. And from that alignment, we will get nodes in our supergraph. So these nodes are coming from the backbone alignments of the cosine-ish functions, while these nodes are coming from the backbone alignments of the sine-ish functions. And there are two weights associated with each node. And then from there, we can induce the directed edges. And so we have a directed edge between two nodes as long as that edge existed in either one of the DAGs. And those will also have two weights associated with. And then we can compute our distance by taking the sum of the absolute differences of the weights on each vertex and edge. I guess before moving on, it looked like there is a question.
Yes, exactly. So yeah, I can read out the question for everyone. Could one use DTW as alternative to LP?
As DTW, is that dynamic time warping?
Let's see if Albertus replies. Yes.
Yes. Yeah, so I don't know all the details to dynamic time warping. I know that it is similar to an edit type distance. So I think it is probably, there's some similarities to the backbone distance in some way, but I don't know exactly what all the similarities or differences are.
Great, thank you.
Yeah. Sweet. And then we do have some stability results. We do have a stability result for the backbone distance, which comes from bottlenecks stability for persistence diagrams, but also taking into this additional constraint that we want ordering preserved in our matchings. And then we also have this extremal event DAG stability, which has a lot of hypotheses and symbols in here. But our bound is going to involve the distances, pairwise distances, pairwise infinity distances between functions in our original time series. And that's as well as some different types of constants. And then for the assumption on the local stability, what this means is that the functions that we are matching when computing our supergraph, they have to be close enough to each other in which you can kind of discern that, okay, this min goes to this min, that max goes to that max, but then also one of the functions can have a bunch of little small perturbations to it. Right. Cool. Yes, and for that result, that came from a bunch of building off the backbone stability and a lot of different little interleaving arguments to get there. All right, so then for the rest of the time, I wanted to talk about some of the applications on how we can use extremal event DAGs and distances to answer biological questions. All right. So the first question I'll look at is, can the circadian rhythm genes of a mouse provide evidence for an intrinsic clock in the malaria parasite? Okay. And this question is motivated by the observation that the blood stage cycle and the infection of the malaria parasite, Plasmodium falciparum, exhibits a 48-hour developmental cycle that culminates in the synchronous release of parasites from red blood cells. And this triggers 48-hour fever cycles in the host. And so this cycle could be driven extrinsically by host circadian processes or by a parasite intrinsic oscillator here. And this question is good for extremal event DAGs or any type of time series descriptor that's trying to keep track of orders of mins and maxes. And that's because if there is an intrinsic oscillator, then the order of the mins and maxes across genes should be similar in different strains of this parasite here. And this paper in Science, published a couple of years ago, did answer this question in saying that there is an intrinsic oscillator that drives this blood stage cycle. And they used and to help answer it, they used a directed graph descriptor, keeping track of the orders of mins and maxes and looking at directed graph distances between them to help come to this conclusion. And so I wanted to look at the data from this paper and see if I can reproduce the results and how the extremal event DAGs do in comparison to the methods they used. Okay. So the malaria data consists of 4 strains and the heat maps of the data sets are shown here. So each row is representing a different gene, and then going across is the gene expression. And so we can see the heat maps. And they do look somewhat similar, so we could suspect that these orders of mins and maxes is preserved across these different strains. We also looked at 3 strains of mouse tissues, and the reason why we did that is because mice have known circadian genes. And so looking at different extremal event DAG distances across the different mice tissues can give us an idea of where those distances range when there are known circadian genes. Okay. So for our results, what I did here is first looked at the mouse tissue data, and I picked out 15 or sampled 15 genes from liver, and then sampled the same 15 genes from lung, computed the extremal event DAGs, and then computed the distance and recorded it. And then I repeated that 1,500 times. And so that's what the purple histogram is representing, that distribution of those distances. And then the blue, these are baseline distances. So this is from taking the data sets and then just scrambling up the numbers there. So then we can see where the extremal event DAG distances range if you're just considering any kind of numbers in the data. Nice.
We have a question. How are the genes ordered on the heat map on the y-axis?
That, I am not sure. I would have to look into that.
Thank you.
Yeah. Great. Cool. So I did that for liver and lung, and similarly for liver and kidney. All right, so this gives us an idea where distances range when there are circadian genes. And then if we look at doing the same thing, but for the parasite, we see that these distances are even smaller. So this does provide evidence that there is an intrinsic oscillator driving our fever cycles. Okay. Cool. All right, so this was great, matching the results in the literature. But then also as an added bonus is that these extremal event distances, they can take in more data than what was used in the paper. In particular, here we were looking at 15 genes at a time, and that number was a little arbitrary. I think we could have tried more since these computations were only taking a couple hours. But in the paper, they were only taking in about 5 genes at a time. The extremal event DAG does not have a noise parameter, whereas the previous method, you did have to specify an epsilon noise level and then repeat the experiments for a bunch of different noise levels and figure out how to average and aggregate those together. And then for that, as a result, this extremal event DAG distance can be computed much faster. It also doesn't rely on an NP complete graph theory problem, which the previous method did. It relied on finding the maximal common edge subgraph. Sweet. All right, so that was all good news. And now for our second application, this is what we are currently working on, where we want to apply extremal event DAGs to yeast cell cycle data to find a gene regulatory network for Cryptococcus neoformans. So Saccharomyces cerevisiae, shown on the left, and Cryptococcus neoformans, shown on the right. These are both different yeast organisms. Cerevisiae is used in brewing beer and baking, and it's been very well studied. People know what this gene regulatory network looks like and which genes are controlling the cell cycle. But for Cryptococcus, it's not as known as what genes are driving the cell cycle there. And despite evolutionary divergence, though, it's been found that these two budding yeasts have a similar percentage of genes, about 20%, that is periodically expressed during the cell cycle. And in particular, genes regulating DNA replication and mitosis exhibited a conserved ordering in both yeasts, suggesting that essential cell cycle processes are conserved in periodicity and timing of expression.
Robin, could you remind us what the scale for the heat map represents?
Yeah, so for the scale, it represents the value of the gene expression. And so you can think of that as gene level activity or how proteins are being produced.
Thanks.
Awesome, thank you. Great. All right. Yeah, so we want to find a gene regulatory network for Cryptococcus, and we hypothesized that the network topology of this gene regulatory network is similar to the well-studied gene regulatory network for Saccharomyces cerevisiae. And additionally, we assume that the transcription factors of the genes for the two networks are pretty similar. Right. Cool. Just going a little into time series and regulatory networks is that time series provides a discrete measurement of dynamical system, and we can infer potential causal relationships between components from a regulatory network. So in this example here, the SWI4, YOX1, HCM1, and NDD1, these are genes in Cerevisiae that are controlling the cell cycle. And here's a nice regulatory network. And so it's noting is that if we, as the level of activity of SWI4 is going to increase the activity of YOX1, and then YOX1 will then repress SWI4. All right, so that's how we can read these types of arrows in this regulatory network. And then on the left here, these are time series data of those 4 genes. All right. And often, many people are interested in associating a dynamical system to a regulatory network to better understand how all these genes are interacting with one another. And there's been a lot of work done in this regard. One is with dynamic signatures generated by regulatory networks, DSGRN. So this is a method to describe the global dynamics of a regulatory network for all parameterizations of a switching system, where a switching system is an ODE. So here's an example where you have your regulatory network. This is one particular switching system that you could associate to it. And there's lots and lots that we could associate it. So that's where the different parameterizations come into play, is the different types of switching systems we can have. Here's a phase space of the switching system describing the dynamics. A state transition graph can be produced, which is summarizing the phase space. In particular, each of these cell blocks corresponds to a node or vertex, and then the arrows are summarizing like where the trajectories are going from one cell block to another. And this search graph is telling where the mins and maxes could occur in the system. And so there's different symbols here, but these capital M's are denoting maxes and the little m's are denoting mins. So it's saying that we could have, or say, a min in X1, and then a min in X2, and then a max in X1, and then a max in X2. That's what the search graph is saying. And then we can compare DSGRN, the dynamics of the network, to the data. And we're often interested in identifying whether our model for dynamics is capable of producing sequences of maxima and minima that do not contradict the experimental data. So for in this example here, here's our phase space. Here's a search graph that this all comes out from DSGRN. And then we could also look at our data and a post set describing the ordering of the extrema. And then there's a way to construct this pattern graph. So it's describing the ordering in a consistent manner to the search graph. And we can look at paths within this pattern match, within this pattern graph, to see the sequences of mins or maxes align with the search graph. And so in this example, we have kind of the sequence of mins and maxes here that matches up with what we found in the search graph. So it could be saying that the model for the network could work potentially. Okay, so then our approach to regulatory network finding is that we use a combination of these extremal event DAG distances, backbone distances, and LP distances to find good gene candidates for controlling the cell cycle in Cryptococcus. And one thing that we do is we look at our Cerevisiae genes, and here we're finding the 10 genes in Cryptococcus that are closest to SWI4 according to the backbone distance, but we've also checked out different LP distances here. All right. And then these are similarly top 10 genes that are closest to HCM1 in the backbone distance. Okay. And then for our network is we want to assign, one of these CNAGs to SWI4, another one of these, a different one of these CNAGs to HCM1, and so forth. However, which ones do we pick out? In order to try to determine that, that's when we apply our extremal event DAG distance. So we look at all different sorts of combinations of selecting genes from these pools. And then we compute extremal event DAG distances and create this histogram here. And what's shown below here is that the extremal event DAG distance between this collection of genes and then this bottom collection of genes from Cryptococcus gives us the smallest extremal event DAG distance. And so this is going to be our candidate network to check out for Cryptococcus. All right. Great. All right. So we have our gene candidates. And then for our second bullet point is that using the same network topology as the well-studied Cerevisiae, we use gene candidates and DSGRN to assess how well the dynamics of the proposed network for Cryptococcus aligns with the dynamics of the time series. And then assessing and seeing how many kind of matches in the mins and maxes we get, we can then start to rate these different types of networks and then propose these network candidates to biologists who can then run experiments to assess the proposed networks and see how sound they are biologically. All right, and so that's what we're in the middle of doing right now. So in summary, extremal event DAGs are a descriptor for multidimensional time series data that is robust to discrete time measurement noise. And the extremal event DAG distance can be computed and has some stability properties. And the biological applications include comparing model organisms to non-model organisms to answer questions involving circadian rhythms and genes controlling the cell cycle. Most of the work I presented can be found in our paper, Extremal Event Graphs, a stable tool for analyzing noisy time series. And we do have code on GitHub that can compute these extremal event DAGs from data as well as distances. And with that, I will end there. Thank you so much for your time.
Thank you so much, Robin. So I'd ask everyone to unmute themselves so that we can give a round of applause for Robin. Okay, and now it's time for questions.
I would be curious to hear a bit more. It's a bit of a vague question, but how did you decide for this DAG, the directed acyclic graph? Like in the sense, would there also have been other methods to describe the information about the order of different extrema? Like for example, maybe just a list of one extremer after another, or I don't know, just I guess you have put a lot of thought into this, and I didn't.
Yeah, like how the project came about.
Yeah, kind of like, or why did you describe for a directed graph instead of some other way of describing this information? If you have some interesting thoughts about this.
Yeah. No, this work follows up from previous work that just looked at like a post set for describing the ordering of the mins and maxes across all the different time series. But then the main motivation was trying to like, how do we take into account noise? And so previously it's like you can create a post set for each different noise epsilon parameter. And then, then you can try to make conclusions from there, but often you don't know the noise, so you have to check a bunch of them. So we were trying to figure out like, how can we create some sort of descriptor that takes into account like all the epsilons somehow. So that's where the weights came into play in creating this weighted directed graph descriptor.
Okay, cool, thanks.
Yeah, thank you.
And then we have Nikki Sanderson who raised their hand.
Hey, Robin.
Hi, Nikki.
My question is about the nature of the time series that you're looking at to measure the differences in. This seems like you want like a continuous time series. Is that like an assumption that you're making?
Yeah, no, I did describe the construction using continuous time series, but we do work with discrete data so that the gene expression is only taking at a finite number of times. Okay,
could I follow up a little bit?
Yeah.
Like like neuron spiking timing is something that's sometimes really hard to kind of, be confident about the order that the neurons are firing. And sometimes there it's just like 0, 0, 0, 1, it's spiked. Is that something that this method could work with, or would that be a challenge?
Oh, possibly. Yeah, I would have to hear more about your data and how, and what it looks like. But that would be cool. Yeah, I'd be happy to talk more about that at some point.
Okay, thanks. Appreciate it.
Yeah.
Thank you. And now we have a question from Georg.
Hi, thank you so much. Very, very interesting work. In a way, like my question, I guess, segues into the previous question, because I think early on you mentioned that you're excluding flats as extrema. Have you what are you doing when you, because in natural data you can get a flat, right? It can happen. Do you do any pre-processing, or what's your, this is a very applied question, I'm sorry, but what's your approach to deal with that sort of exception you're making?
Yeah, no, that's a good question. And I think in our data, I haven't come across this where we have like a flat max or a flat min. I think what first comes to mind is just trying to perturb the data a little bit somehow, like maybe just moving one of those data points
Up and down a little
up
bit,
a little bit. Yeah. And then
Yeah.
you would be good. But yeah, that's a good thing to think about.
All right. Thanks so much. Very nice. Thank you.
Yeah.
More questions? Go ahead.
Hi, Robin. Great work. I have a question about the properties of the time series. How this method apply for a missing time series with missing data, or time series of disconnected components. Sometimes there's the measurement, sometimes there aren't, like irregular type of time series.
Yeah, so for this method, it's fine to use, like if not all the time series have the same number of time points, that's okay. But I guess for the noise that we're talking about, it's not so much about the noise with having like missing time points. So I feel like if you want to take that into account, you'd have to incorporate some other methods.
Thank you.
Yeah, thank you.
Thank you. Do we have more questions for Robin? Okay, so then I will stop the recording and we can take questions offline. And Robin, thanks so much again for your wonderful talk.
Yeah, thank you.